Welcome student on another video lecture. Topic is biosynthetic pathway, and in that the last technique we are going to study, that is tracer technique, which is used to find out or trace the different intermediate and to trace the biosynthetic pathway which the plants will follow to synthesize various type of secondary metabolites. So what is meant by tracer technique? It is defined as a technique which utilizes a labeled compound. So in this technique, label compound is used, and that label compound will trace the different intermediates which are formed during various steps of chemical reaction, which are synthesized by or which are catalyzed by different enzymes. The intermediates are produced, and that intermediates are traced by label compound in various steps in biosynthetic pathway in plant at a rate and at a time. So at particular rate or at particular time, these intermediates are produced during various biosynthetic pathways which the plant will utilize to synthesize secondary metabolites. So this technique uses a label compound, and that label compound will trace these intermediates. Label compound can be prepared by use of two types of isotopes. So the label compound which is used to trace that intermediate, they are of two types. First one is radioactive isotope and second one is stable isotope. Now radioactive isotopes, the examples of radioactive isotopes are carbon 14, phosphorus 32, hydrogen 1, sodium 24, sulfur 32, phosphorus 35. All these are the different radioactive isotopes which are used to trace the intermediate. For biological investigation, carbon and hydrogen isotopes are used. So to do some biological investigation, then carbon and hydrogen isotopes are used. For metabolic studies, sulfur, phosphorus, alkali, alkaline earth metals, all these are the compounds which are used for metabolic studies. And for studies on proteins and amino acid, then label nitrogen radioactive isotope is used. So nitrogen, labeled nitrogen is used for protein and amino acid. Metabolic studies, sulfur, phosphorus, alkali, alkaline earth metals, all these are used as a radioactive isotopes. Now second category is stable isotope. Example is hydrogen 22, carbon 13, nitrogen 15 and oxygen 18. These are scarcely available in nature, used for labeling compound as possible intermediates in biosynthetic pathways. So these are used as a labeling compound and to trace the intermediates in biosynthetic pathways, and which are detected by mass spectroscopy or NMR, nuclear magnetic resonance spectroscopy. These techniques are used for its detection. Now significance of tracer technique. Tracing of biosynthetic pathway by incorporation of radioactive isotope to the precursor or starting material, or the whole biosynthetic pathway can be traced. So what is the significance of tracer technique? First significance is that by incorporation of radioactive isotope into the precursor or starting material, when the radioactive isotope, that is label compound, which is inserted into the precursor or starting material, then it will helpful to trace the complete biosynthetic pathway. For example, by incorporation of radioactive isotope of carbon 14 into phenylalanine, so here the radioactive isotope, that is carbon 14, that is incorporated into phenylalanine. It is a amino acid. The biosynthesis of cyanogenetic glycoside pronacin can be traced. So that from that phenylalanine, that is amino acid, from that amino acid the secondary metabolites, that is prunicin, which is a cyanogenetic glycoside, the biosynthesis of that prunicin can be traced. Second one is location and quantity of compound containing tracer. If location and quantity of glucose is determined in a biological system, the carbon 14 labeled glucose may be used. Second significance is that location and quantity of compound containing tracer. It will helpful for location and how much compound and the quantity of compound which that is available in the trace containing tracer, that is also find out. If location and quantity of glucose is determined in biological system, the carbon 14 labeled glucose may be used. So here the isotope, that is carbon 14 labeled glucose isotope, is used to find out the location and quantity of glucose. The labeled glucose being chemically indistinguishable from the native glucose. So native glucose is different and the labeled glucose is different. Will mix completely with available glucose poles in the body of organism studied, and it will mix completely with the glucose poles in the body of organism which is under study. Both location and quantity of glucose present in tissue can be determined by radioactive assay. So how much, what is the location of that glucose and how much quantity of glucose that is present in the tissue, that can be determined by radioactive assay. This is the significance. After that, different tracers for different studies. For studies on protein, alkaloids, amino acid, label nitrogen atom give more specific information that label carbon. So if we have to study the proteins, alkaloids and amino acid, then nitrogen atom is more specific and it will give more information as compared to carbon. So if the studies on protein or amino acid that are carrying out, then nitrogen, labeled nitrogen is generally used. So all these three are the significance of tracer technique. Now criteria for tracer technique. The starting concentration must be sufficient so as to withstand resistance with the dilution in course of metabolism. So when the starting concentration is proper, then because when that tracer compound, label compound will undergo metabolism, then dilution will occur. So the concentration should be sufficient so that it can withstand the dilution in course of metabolism. Label compound must be involved in synthesis reaction. That label compound should also involve in the synthesis reaction, then only we can trace the biosynthetic pathway. The label compound must be harmless to the system to which it is used. So whatever the label compound or radioactive isotope it is used, that should be harmless. It should not cause any harm to the organism on which the study is carried out or the plant on which the study is carried out. So for that reason it should be harmless. The proper labeling is required. For proper labeling, physical and chemical nature of compound must be known. Whenever the label compound is inserted into the precursor, then proper labeling is required, and the chemical and as well as physical nature of that compound must be known before inserting into the precursor. The tracer should be highly pure. The tracer compound which is used, that should be pure in nature. The radioactive isotope with greater half-life period is preferred, because if the half-life is more, then the radioactive isotope will remain in body or in organism for longer period of time, and that will be benefited for tracing the biosynthetic pathway. If the half-life is very small, then the compound will not remain longer period in the organism, and it is difficult to trace the biosynthetic pathway. For that reason, the greater half-life period should be preferred. For example, carbon 10, carbon 11 and 11 carbon, it is having 8.8 second to 20 minutes half-life, and carbon 14 about 5000 years. So it is carbon 14 is mostly preferred because of its longer half-life period. So this is about the criteria for tracer technique. Now what is the method and how the tracer techniques is performed, that we will see in our next lecture. So if you like my video, please press the like button and subscribe my channel. Thank you.